Good morning, good afternoon, or good evening, depending on where in the world you are tuning in. I'm Max Häggblom, Editor-in-Chief of FEMS Microbiology Ecology, and it is my pleasure to welcome you to this webinar on aquatic microbial ecology. FEMS continues the series of webinars to support the microbiology community. As a not-for-profit organization, FEMS, the Federation of European Microbiological Societies, uses the income from our journals to fund our charitable activities and support our communities. FEMS journals invest in science. We provide grants to scientists every year, present several prestigious awards, organize and support conferences, and sponsor a range of events such as this webinar series. These webinars provide a forum for the presentation and discussion of key research, enabling the flow of ideas to continue despite the cancellation of in-person events and conferences. Each month, we highlight a different topic of microbial ecology. And if you missed our earlier webinars, they are available on the FEMS YouTube channel. Today, we focus on the fascinating topic of aquatic microbial ecology. Aquatic habitats are rich environments for microbial life and have global impacts on biogeochemical cycles. The behavior of microbes in response to changes in these environments can have a great influence on ecology across the world. The topic will be introduced by our editor, Martin Hahn, followed by presentations by Marian Schmidt, Megan Dillon, and Wei-Dong Kong, who explore three different aquatic habitats: a freshwater lake in Michigan, Lake Frixell in Antarctica, and Tibetan Plateau lakes. After the talks, we will open the session for question and discussion. You can submit your questions via the question link. So to begin, it's my pleasure to introduce Martin Hahn from the Research Department of Limnology, University of Innsbruck, who is also editor in FEMS Microbiology Ecology, handling a range of papers in aquatic microbial ecology. And Martin, I'll give the floor to you.
Thank you, Max. Hope you can hear me. So I switch to the full screen. Oops. So I think everybody can now see this screen. So since today's topics of the three papers presented later is on inland waters, I will give a brief overview on the diversity of inland waters. So in the title, freshwater was mentioned, but we should better consider inland waters here. And I just want to emphasize how incredibly diverse inland waters are. I have here some photos. So there are no saline systems shown, no running waters, but you already can see, I hope, how diverse freshwater systems are. They differ in size, in depth, in stability, which is somehow related to the environmental influence from the terrestrial influence, or by the size itself, different in chemistry, nutrients, and connectivity to the environment and to other freshwater systems. And there are a lot of different terms which all highlight these differences. So freshwater, saline, standing waters, running waters, lakes, ponds, puddles, and so on. So we have a really huge diversity. Also in terms of chemical properties of the different systems. Here we see a graph showing the salinity of some example systems and the pH. And as you can see, pH may range, not shown here, but it may range from below 4 in inland waters to more than 10. And the salinity, if we include saline lakes, then is going beyond salinity of the oceans. So really huge chemical diversity. And today we will, as as here I am. So as Max already mentioned, we will hear different examples from different systems, freshwater lakes, oligosaline systems, and a broad range of lakes located on the Tibetan Plateau. So as mentioned, pH, salinity, major ions, and all this is making chemical differences in the systems. And this is, I believe, quite well shown in this satellite image of the confluence of Rio Negro and the Amazon River. There you can see how different the water quality, the water chemistry of, in this case, freshwater systems can be. They differ, these two rivers, they differ in pH, they differ in the organic load or inorganic load, in conductivity, and so on. Not always it is so easily visible how different the chemistry of the water is, but again, we have a very wide range. When it comes to systems, then you have these textbook examples of lakes stratifying and then turning over, for example, in fall and in spring. Then you have what textbooks call dimictic lakes. There are other lake types, monomictic lakes, polymictic lakes. So lakes can be quite different in their thermal stability, or they can be stratified by chemical gradients. In the case of meromictic lakes, we will later hear about an example in one of the talks. But if you look on some specific habitats, individual habitats, then it's pretty well clear that not all freshwater systems, standing freshwater systems, follow simply these textbook schemes. Here I have an example from a shallow lake, a small pond actually, and this is only 1.6 meters deep, so really shallow. And I have here some temperature data ranging from spring to mid-summer. The temperature was measured in different depths. So here you can see the different depths. Blue is, it started in 1 meter and 45 centimeters, but then the lake water level decreased. So at the end of the measure period, the depth was only 1 meter and 25 centimeters. But don't be confused about that. Look on the blue curve. You can easily see that in this deeper water, a little bit more than 1 meter, the temperature over the days was more stable. If you look on the measured temperature closer to the surface, so in the beginning 25 centimeters, this is the red color, you can see how strong the temperature is going up and down each day, cooling during the night. Stratification, yes, it took place for longer periods. If you look on this part of the graph, but then it broke down, not in fall, but in May. So this is just an example how complex the conditions in a particular habitat can be. And then it comes to microhabitats in addition. So not all the microbes live under the same conditions in a particular habitat. So some are host associated, others live as planktonic cells, others are attached, some live in deeper waters, and so on. So a lot of different conditions in one habitat. And given that huge diversity of inland freshwater, of inland systems, we have to be aware of the fact that it is very difficult to identify universal mechanisms which take place, for example, in microbial community composition or assembly, diversity, ecosystem services, and so on, which take place across this huge diversity. So can we find mechanisms, can we identify mechanisms really valid for all these different systems, or do we have to search for groups of systems, habitats, or microhabitats, which are then controlled in some of these parameters by common mechanisms, but distinct mechanism may then take place or may control in other systems. So I believe it's really quite difficult to get the whole picture. Now I switched wrongly, so here I'm back. To get the whole picture across all this diversity, and we have to ask how far we came so far with our research and our understanding across this diversity. Thank you.
Thank you, Martin, for the introduction, and we'll definitely see how over the rest of the webinar how varied these systems are. And so it's my pleasure to introduce our next speaker, Marian Schmidt from the University of Texas at Austin, who's going to talk about microhabitats in freshwater bacterial communities. So you should have it. Are you still muted?
Hello, can you hear me now?
Yes.
Yes, okay, great. Hi, everybody. I'm really excited to be here today. And, ooh, there we go. Currently, I'm a postdoctoral fellow at the Department of Integrative Biology down here at the University of Texas at Austin. I'm currently focused on some evolutionary aspects of microbial communities, but today I'm going to be sharing some of the work that I did during my PhD at the University of Michigan. And I'll specifically be talking about how microhabitats are associated with diversity productivity relationships in freshwater bacterial communities. And since this is a little bit of a unique situation where I've already published the paper, I want to take a moment to recognize the colleagues that collaborated with me on this project. And so when I was a PhD student at the University of Michigan, I worked with Vincent Deneff. And so this was a collaboration between the Deneff lab and Dr. Bopi Bidanda's lab at Grand Valley State University. And I had some funding from the National Science Foundation and the University of Michigan, and the Margaret Ayers Host Award, which recognizes women in science. So we focused our study on freshwater systems, and specifically these ecosystems, as Martin just pointed out, are very diverse, but we also know that these ecosystems are changing, and they're changing both due to climate change and also due to human perturbations. And one example of this is eutrophication that's fueled mostly by agricultural runoff. And so here we can see a famous study done where they manipulated the amount of phosphorus in one side of the lake, and we can see there's a huge amount of biomass that's present on one side of the lake, indicating eutrophication. A process that's also very related to this is the harmful algal blooms. And we know that harmful algal blooms are becoming much more of a problem in freshwater ecosystems. And currently where I live in Austin, these harmful algal blooms can actually span around 6 months of the year. And so they can have significant negative impacts on the organisms that live on these lakes or depend on these lakes. In addition, storm runoff is becoming more intense because with climate change, we expect for storms to become more intense and more frequent. So here's an example also from here in Texas, where there is a pre-storm here. We have a river, and then after the storm, there's a huge flooding event that brings even entire tree trunks down into this system. And so these storm events actually increase the linkage between the terrestrial environment and these aquatic ecosystems. And it's a one-way linkage in this case. In addition, species invasions are becoming much more important in freshwater systems. And the region in which I study, the Great Lakes region, it's in a continuous state of invasional meltdown, where there's an invasive species after invasive species. Finally, we have microplastics that are also making their way through our wastewater treatment plants. And the impact that they have on these freshwater ecosystems is mostly unknown, although some really interesting research has been coming out. One thing that I'm really interested in, as Martin pointed out, is that there's microhabitats that microbes can live on. And so one thing that all of these different climate and human perturbations have in common is that they influence the type and the amount of particles in these ecosystems. Most of them, as we can see by the red arrows, increase the number of particles in these systems, but in the case of species invasions, here the dressed mussels, we might expect for them, their filter feeders, to decrease the number of particles. But what might we lose or gain due to these changes in the amount of particles in these systems? Well, they have some important ecological contributions. And so while the particle organic carbon pool is 10 times smaller, it's much more bioavailable for microbial communities. In addition, in oxygen minimum zones in the ocean, people have noted that nitrate reduction to nitrite only takes place in particles. And I think that the application for this to anoxic or microtoxic lakes in the bottom of the lake could be really interesting. In addition, microbes on particles have a wider array of polysaccharide hydrolase activities, meaning they could also disproportionately contribute to carbon cycling in these systems. And some cool work done on model marine particles shows that there's a rapid species turnover or ecological succession of the microbial taxa that eat and feed off of one another on these, where there's primary degraders that first colonize the particles. Later on, there's secondary degraders that feed off the byproducts of those primary degraders. And then later on, there can be cross-feeding and cheating that happens. And so there's some really interesting biogeochemical and ecological interactions that takes place on these particles. And so zooming into a parcel of water collected by my collaborator, Bopi Bidanda, we can view these microhabitats more directly. And so a definite oversimplification of this view could be that there's free living microbes that live alone solely in the water as plankton. And then there's other bacteria that live in association with other cells. And these particles act as microbial meeting points. And so here, for example, this large, relatively pennate diatom is housing a home for these microbes that are likely feeding off of the extra photosynthate. And so it's creating this microhabitat. And so today I'm going to be talking about testing some ideas for microbial ecology in this system. And I first am going to ask whether or not particle microhabitats support more diverse communities than the free water that's literally surrounding it. Second, I'm interested in asking, how does bacterial heterotrophic production vary between these microhabitats? And then finally, linking those together, are there biodiversity ecosystem function relationships in lake microhabitats? And if you're like me, when I started graduate school, I had no idea what biodiversity ecosystem function was because I was trained as a microbiologist. And so this theory posits that the number of species that performs a process is likely to be related in some way to ecosystem function. And one thing that is so cool about using this theory and applying it to microbial systems is that there's a vast metabolic diversity that microbes have. So this theory was first developed in plants to study the ecosystem function of plant biomass, because perhaps as we lose extinctions of species, might we see, for example, if we cut down forests, a difference in the amount of plant biomass. And here we might expect different things to be, or different relationships based on different types of functions like methanogenesis, sulfate reduction, or denitrification. And so you might think about your favorite microbial metabolism in this case. So this relationship might also depend on the number of taxa that perform that process. And so over here on the left side of the function, we have little functional redundancy or a lot of functional redundancy on the right side. So we might expect that for processes performed by few taxa, that there's likely to be a relationship that's positive, because what's actually limiting the function is literally the abundance and the number of species that perform that process. However, once we get over here to the right side of the function, we might expect there to be no relationship by functions performed by many taxa. For example, like cellular respiration. And that's because now the environment can be the limiting factor, and it's not due to abundance or taxa present. So I'm interested in how this side of the story might, I'm interested in this half because I'm really interested in how heterotrophic production might be related to diversity function. And that's a process that's performed by many freshwater bacteria. And I'm specifically interested in how might particles modify this relationship. So today I hypothesize that particles influence the strength of this diversity function relationship. And there's a few different ways that we can test this in microbial systems. First, we could do, for example, a dilution to extinction where we are removing taxa from the community. We could perform manual assembly of communities where we take cultures and we put them together and modify the diversity that way. Or we could use natural or manipulated environmental communities, which is the approach that I'm going to show you today. So, as Martin mentioned, I went and I studied Muskegon Lake here in the state of Michigan in the Great Lakes region, and it's found on the western side of the state. And actually, it's an ecosystem that's kind of represented along the entire shoreline of the state of Michigan, right next to Lake Michigan here. And it drains a very large watershed that's around 3,000 square miles and has a mix of agriculture, forest, open field, and urban landscapes. And that watershed flows into this lake on the east side from the Muskegon River. And over around 3 weeks, the water makes its way out of Muskegon Lake and into Lake Michigan, one of the Great Lakes. And so what we did was we took advantage of this type of productivity gradient. And so we sampled near the river, near the inlet to this other lake, Bear Lake, which is a eutrophic lake. We took samples at the deepest site and at the channel, all at 1 meter depth. And I had some pretty amazing field assistants that came with me for the long days on the boat. So what we did was we took lake water and we first filtered it onto a 3 micron fraction. And then what went through that onto a 0.22 micron fraction, we called the free living. And I'd like to point out that this is not necessarily the best measure. It is a nominal cutoff that is pretty common these days in the literature. And so I want to point out that there could be, for example, large microbial cells that end up on the 3 micron fraction. And so it doesn't necessarily mean that they're all particle associated, but likely the vast majority will be. And then finally, we performed DNA extraction, 16S gene sequencing, and then measured diversity using this R package called iNext. And so how does the diversity of particle communities compare to the free? Before I jump into that, I'd like to discuss briefly, how does one measure diversity? When I entered my PhD program in ecology and evolution, I thought I really understood what diversity meant, but in reality, I didn't. And so I just want to clarify what I mean today about it. And so this is within sample diversity or alpha diversity. And I'm going to be talking about the Hill diversity metrics. And these metrics were formulated to make cross comparisons between studies much more feasible, to kind of decrease the issues related to rarefaction and so on. And so it differentially weights species richness and relative abundances with the effective number of species using the order Q. So as Q increases from 0 to 1 to 2, the species abundances are weighted more. And that also decreases the sensitivity to sampling effort. And all of this is provided in one single equation where we can modify Q. And as it's 0, richness is weighted more, but as it's 2, the species abundances are weighted more. And this gives a really interpretable diversity measure. Essentially, it provides D0, which is the effective number of observed or total species, D1, which is the effective number of typical species, and that's mathematically equivalent to the exponential of the Shannon index. And then third, there's the effective number of dominant species, which is equivalent to inverse Simpson. And so going back to the question that I was asking, how does diversity of particle communities compare to the free? So what I found was that when richness is weighted more heavily, particle communities are more diverse. So here we have the effective number of total taxa and also typical taxa. However, when we weighted evenness more, what we saw was that there was no difference between the number of dominant taxa. However, I would like to note that there's much larger variation in the particle-associated communities, and this is predominantly driven by the springtime. But what about this over time? I mentioned that we sampled it at different seasons. So now I'm going to show you the difference where I take the particle diversity and I subtract the free living diversity from it. And so it'll show kind of a difference over time. And so here is the number of observed taxa. And so when it's in the red zone, that means particles are more diverse. But if it's in the yellow zone down here, then the free living is more diverse. And so what we can see is that especially in the springtime, particle communities are more diverse than the free living, but not for the fall. And so they become much more similar later on in the season. And we can actually see that this is being heavily weighted by the two lake sampling stations by the river and Bear Lake right here. When we weight the more dominant taxa, what we can see is that there still is seemingly a trend, but this trend is insignificant. But we can see that the fall tends to have more diversity in the free living compared to the particle. Okay, so how does bacterial heterotrophic production vary between particles and free living? So I've talked about the diversity side of the story, and now I'd like to talk a little bit about the productivity side of the story. And freshwater systems help to regulate the global carbon cycle. And so there's this idea of the active pump, where around 3 petagrams of carbon per year enters inland waters, and about a third of it makes it out into the oceans. Around half of it is respired out into the atmosphere, and then a smaller amount of it is buried in the sediments. And I was curious, like, what do these enormously large numbers truly mean? And specifically, I was interested on this atmospheric component, because heterotrophic bacteria in these inland waters are the predominant producer of this carbon that is being released. Of course, there are other organisms that also contribute to it, but due to their sheer abundance, heterotrophic bacteria are the predominant contributors. So if we take 1 petagram, which is around 10 to the 15 grams, and we do kind of a fun calculation, and if a blue whale is the largest organism on the planet, how many blue whales worth of carbon might these heterotrophic bacteria in inland waters respire annually? So that's around 14 million blue whales worth of carbon every year. And that's 2 blue whales around every 5 seconds, which is the amount of time that it takes these blue whales to swim across my screen. So we just lost 2 blue whales worth of carbon due to microbes in fresh waters, inland water. Okay, so I didn't measure this directly, but instead what we did was we took an approach to measure it indirectly. And so here we measured heterotrophic bacterial production, and we did it by using the method of a tritiated leucine incorporation analysis. And this is a proxy for heterotrophic bacterial biomass, which is related to the amount of respiration that happens. So slightly different. Whoop. Okay, so how many cells are in each habitat, and what is their functional role? And so through doing some kind of microscopy counts of looking at what was associated with particles and what was free. So of course there's some human error in that. 10 times more cells per milliliter in the free living. And when we looked at the tritiated leucine incorporation analysis, what we found was that there was more total production in the free living, which was unsurprising because we know that there's around 10 times more cells. However, when we back calculated this and we asked what about the per capita or per cell production happening in these two microhabitats, what we saw was that particle cells are disproportionately active compared to those in the free living. And so there's more production per cell in the particles. Okay, but going back to my larger question of does bacterial diversity influence ecosystem function in microhabitats, looking at heterotrophic production. So here I'm going to show you the effective number of observed taxa on the X, or D0, and the total heterotrophic production. And what we found was that there was a diversity productivity relationship, and it explained around 60% of the variation in the data, but it was only present in the particle communities. And a cool thought experiment to perform is to look at the x-intercept. And so here we can see that the x-intercept is around 406 taxa. And here I'm wondering, oh, maybe perhaps this is a baseline level of the number of inactive taxa. Perhaps they could be dead, they could be dormant, perhaps it's environmental DNA, or taxa that are actually unable to incorporate leucine, because we know that there is definitely a bias in that. Okay, but what about when we weight evenness more or look at the number of dominant taxa? So here, if it's higher, that means it's more even or there's more dominant taxa, and looking at total production. And what we saw was that this relationship actually strengthened as we weighted the dominant taxa more. And so now we were able to explain around 70% of the variation in the data. But you might be wondering, I went out and I sampled environmental communities that live in nature. So perhaps I could just have some environmental correlates that could be more important factors than biodiversity in this system. So what about the environmental variables? Does the environment or biodiversity better predict heterotrophic production? So to get at this question, what I did was I performed a lasso regression, which allows me to perform variable selection. And so it penalizes variables that doesn't inform what we're trying to predict, in this case, heterotrophic production. And so it shrinks down and removes those variables and only leaves the ones that are the best predictors. So when I did this, what I found was that the particle heterotrophic production was best explained by biodiversity, specifically the total number of observed taxa and the number of dominant taxa. But in the free living, the environment was the best predictor of heterotrophic production. And in this case, it was pH. So we can see that for the microhabitats of particles, biodiversity matters the most, but the environment is more important for the plankton. So if we revisit the schematic I mentioned earlier in my talk, where there was supposed to be no relationship with functions performed by many taxa, what we actually saw that maybe it's both the number of taxa and the habitat that determines this relationship. So particles have much more heterogeneous habitats. And so that could be the reason why it could potentially extend into this more functionally redundant space. But in the free living fractions, they're much more homogeneous. And this could perhaps be because the source of particles is really unique. So we could have terrestrial particles coming in from the land. There could be photosynthetic or detrital-based particles present in the system. Also, within a singular particle, there could be gradients of nutrients that could support niche partitioning of different types of microbial taxa. And I think that this could increase the likelihood for niche complementarity to occur between taxa on microhabitats. And what I think could be really interesting is that more interactions could be occurring between these types of cells. They're much more close in space, and they have a higher likelihood of, for example, cross-feeding off of one another or sharing genes. Oops, I have a slow network connection. I hope y'all can hear me. But for the free living, it's unlikely that cells will see other cells and that they're going to be influenced the most by the same bulk water conditions. And so things that metabolites they might produce will dissolve out into the water and might make it more unlikely to have these clear interactions with other cells. So in conclusion, I'd like you to walk away with 3 main points. And that's first, that bacterial communities on particles tend to be much more diverse, especially in the spring and near inputs to the lake where the terrestrial environment has, or immigration events have more of an influence on the community. Second, that bacterial cells on particles are more productive. And third, that biodiversity is the best predictor of heterotrophic production in particles, whereas pH was the best predictor of production in the free living environment. And so with that, I am finished, and I guess we'll hold questions to the end, but I did put my email and Twitter on here if you'd like to get in touch. And I'm really excited for the next talk in the series. Thank you for listening.
Thank you, Marian, very much. Really interesting, lots of questions, but indeed we'll come back to these then at the end of the webinar. And so we will move from Michigan via Texas to California and Antarctica. Our next speaker, Megan Dillon from Novozymes in Davis, California, will talk about benthic microbial mats in Lake Frixell, Antarctica. And Megan, I think you have the floor.
Thank you. Hello, everyone, and thank you for inviting me to participate in this panel discussion. So today I'm going to be talking about Lake Frixell, which I studied for my PhD, and a little bit about the principles of microbial ecology and how they relate to some ecological theory that was developed using plants and animals. This work was supported by a global team of collaborators, including Dawn Sumner, who was my advisor for this project. She's been working in the McMurdo Dry Valley Lakes now for about 10 years. This is my co-advisor, Jonathan Eisen, who's been a leader in the field of metagenomics and microbial ecology methods development. We also were very happy to have Anna Youngblood from the Natural History Museum, who is an expert in cyanobacteria. This is Peter Doran, who has been diving for research diving in the McMurdo Dry Valley Lakes and pioneering methods for that. In the water here is Tyler Mackey, my then lab mate and now a professor at the University of New Mexico. This is Ian Hawes, who has been to Antarctica possibly more than any other human on the planet. And over here is Sydney Sally, the newest member of Dawn's research team, and she picked up some of the work that I left off of after I graduated. She's looking at nitrogen cycling in the lakes. We were very fortunate to have some great funding from NSF, NASA, Antarctica, New Zealand, and the support of the McMurdo Dry Valley long-term ecological research team. Antarctica is a very special place. The continent is surrounded by the Southern Ocean, which is very feisty and does a great job of isolating Antarctica from the plant and animal life on the rest of the globe. If seeds or animals do manage to make it to the continent, they're faced with enormous barriers to colonization because most of Antarctica is covered in snow and ice. The McMurdo Dry Valleys are the largest extent on the land that is free of snow and ice, but it is still freezing cold and there's very little precipitation, so these are frozen deserts. It's easy to see why the lakes in the dry valleys act as oases here, because there's liquid water under the perennial ice covers in these lakes. This is a picture of Lake Frixell, the lake that I studied. Like most of the lakes in the dry valleys, it has a thick ice cover. Here it's about 4.5 meters. And this is important for the entire ecosystem. It regulates the organisms growing in the water column and the microbiome that grows at the bottom surface. And it's important in two specific ways. First, the ice cover restricts gas exchange between the lake water and the atmosphere. So because of this, gases and specifically oxygen build up right under the ice cover, and the shallower waters are supersaturated with oxygen. Then the oxygen diminishes and the waters become anoxic around 9.8 meters. And secondly, the ice cover restricts light penetration into the lake. So about 1% of the incident irradiance makes it through the ice cover, and then that decreases through the water column. And as with generally ice-covered lakes and waters, they attenuate the wavelengths of light so that the light is much bluer, shorter wavelength toward the bottom than it is at the top, just under the ice cover. So these are some diagrams of the chemical conditions in the lake. We can see on the left, here is depth going through the water, and then in orange is photosynthetically active radiation, and in black is oxygen concentration. Blue is conductivity. So we can see that the two dominant gradients in the lake, oxygen concentration and photosynthetically active radiation, are correlated through the water column. Conductivity is pictured here just to illustrate that the water is stably stratified. And so as noted earlier, this is a meromictic lake, so it does not turn over. There's no flow or turbulence. The organisms here are limited by diffusion, so nutrients make it through the water just through Brownian motion. And then on the diagram on the right, we can also see pictured here the slope of the basin. So the bottom surface of the lake is very gradually sloping, and this is important because it gives a lot of physical space for the microorganisms growing at the bottom to inhabit similar conditions to each other. As you move deeper in the lake here, the physical forms and the pigmentation of the microbial mats change. At 9 meters, there are pinnacle shapes. At 9.3 meters, we have this pit and ridge structure and these honeycomb type mats. And at 9.8 meters, the mats are quite flat. And here is some of the nitrogen species that Dawn's newest student has been working on. Just shows the relationship to the oxygen gradient and through the oxycline here. This is a picture of what the microbial mats look like at each of these locations where they were sampled at 9 meters, at 9.3, and 9.8 meters. The microbial mats morphology and colors do change gradually and smoothly as you get deeper and deeper in the lake. And these mats here were sampled because they were most typical examples of their type. So these are sort of end member morphologies here. Additionally, all of the microbial mats in the lake are layered. So you can see a really nice depiction of that here. This is an example from the 9-meter mats. And for scale here, these laser pointers are about 3 centimeters apart, and this is 10 millimeters right here. So all of the microbial mats were sampled layer by layer in replicate when we were down there. In addition to measuring the oxygen concentration through the water column, we took microelectrode profiles of the microbial mats when we were there. So here on the y-axis is the distance from the mat surface, going from about 8 millimeters above the surface to 12 below, and here is the surface of the microbial mat. And on the x-axis is oxygen concentration in milligrams per liter. And notice the break here. So the microbial mats at 9 meters, like I was saying before, they're really in super saturated oxygen concentration. They don't ever see any anoxic conditions. At 9.3 meters, those ridge and pit microbial mats, they live in oxygenated waters, much less concentration than at 9 meters. And then they experience anoxic conditions into the sediment. At 9.8 meters, those flat green mats, they are growing in anoxic waters. And this little whiff of oxygen here results from the photosynthesis occurring, the physiological photosynthesis and oxygen that's generated from that by the cyanobacteria that are growing in these microbial mats here. And just 4 millimeters below the surface, so through the mat layers, they become anoxic. So what we have here is this closed basin lake with a couple of dominant environmental gradients, oxygen concentration and light. The microbial mats growing at the bottom, they change in color and structure across these gradients, right? So my first major hypothesis was about how photosynthetically active radiation affects diversity in this system. Because PAR is the major source of energy for this ecosystem, I hypothesized that diversity would be greatest where the primary producers were thriving, because they would be creating more ecological niches. So this is kind of like the trickle-down economics system for microbial habitats. My thinking here was guided by the species energy theory, which states that the more resources there are in a given area, the greater number of species can be supported through niche creation. And so the cyanobacteria, which dominate these ecosystems, they're the net primary producers here. And when I measured diversity, this is what I found. So I'll note here that this is a different diversity metric than was shown previously. This is a phylogenetic measure of diversity. It's abundance weighted, and it's based directly on the branch length between the reads that we recovered with our metagenomic sequencing. Here, these box plots are organized according to depth and layer. So these are the 9-meter mats, the 9.3 meter mats, and the 9.8 meter mats, and then moving from the surface toward the sediment through each of these. So in general, I did find the trend that I hypothesized originally, where there's greater diversity at 9 meters than at 9.3 and 9.8 meters. And that relationship is consistent and it's statistically significant when you group the samples based on their depth. So this trend is significant. But of course, that's not the only trend in these data, right? So there's another trend at any given depth. If you probe from the surface, where PAR is greatest, to deeper toward the sediment, you also see an increase in diversity. And this trend too is statistically significant if you're to group the samples according to their relative positions by layer in the microbial mats from surface toward the sediment. So this contradictory set of trends led me to start thinking a little bit more deeply about some ecological theory, specifically meta-community theory, and the effects of scale. More and more, understanding the spatial and temporal dynamics of any microbiome is increasingly drawing on the principles of ecology that were largely developed using plant and animals to build up their hypotheses and theories. So briefly here, the idea is that local community dynamics, and thus the abundances of specific populations in local habitats and their metabolic output, is dictated by the immigration of those species from a regional pool, what they find upon arrival into their local habitat, and how favorable or unfavorable the conditions are when they get there. Are the neighbors going to be friendly, or are they going to be hostile? These factors are affecting the likelihood of survival and reproduction as we move through the different microhabitats in an environment. So if we want to understand a system, these are all things that we need to consider. In Lake Frixell, we can think of the regional species pool as all of the microorganisms of the lake. The microorganisms growing in the microbial mats at all the depths, and those that are in the water column. We know that some species are motile to various degrees, so there's a lot of gliding motility in the microbial mats. Some of the pelagic community members have flagella, so they do disperse across these ecosystems. And when they migrate from one habitat to another, they may find themselves in an environment that they are not well adapted to, right? So whether that's a different light regime or oxygen concentration than they evolved in or found themselves in originally. So in Frixell, the most important factors here are light and oxygen, and then the ability of immigrants to survive when they hit a particular local habitat has a direct impact on the overall spatial structure of the microbial mats. And then, like I said before, the habitats are also characterized by resident populations. The dynamics of communities then depend on how the immigrants compete with, cooperate with, form trophic interactions with the microbes that are in the local habitats already. So I began to realize that underlying my hypotheses about diversity and photosynthetically active radiation was an assumption about what was controlling the entire spatial distribution of species. Because Lake Frixell is closed basin, there are no outflowing streams, and the input, both in terms of organisms and nutrients, is extremely minimal because of the low productivity of the dry valleys themselves and the glaciers from which the waters are running off. I realized that my actual hypothesis was that niche selection would be a dominant force in this ecosystem, and specifically that because the ice cover limits light penetration into the water column, I was imagining that photosynthetically active radiation was really controlling not just diversity, but which organisms ended up where specifically throughout Lake Frixell. So here I'm showing just a very broad overview of the phylum and family level distributions of some of the organisms we found in the lake. We see that cyanobacteria and these diatoms, Bacillariophyta, both photoautotrophs, are dominant at the surfaces of the mats at all depths. Do notice here that the x-axes are not the same for these three plots. So, but we also notice that the dominance of these photoautotrophs is definitely most prominent at 9.8 meters, where photosynthetically active radiation is much weaker than it is at 9 meters. So this means that PAR cannot be the only environmental feature that's filtering immigrants. And this is where we need to consider oxygen concentration as well. Remember that oxygen concentration is different through the lake and through the mats. Oxygen concentration is supersaturating at 9 meters. So there's really no physiologically meaningful difference between the layers at 9 meters with oxygen. It's the only difference between these layers is with photosynthetically active radiation. So we think here that PAR is controlling the distribution of the species dominantly in this location, in these habitats. At 9.3 meters, because of the morphology of these pits and ridges in this honeycomb mat, Light appears to be penetrating into the pits due to some photography that was done in previous expeditions. And we also know from some work with the microelectrodes that within the pits, the bottoms are anoxic. So there's a local oxycline going through some of these pits here. We also found phototrophs in all 3 of the layers from top to middle to bottom. And there's no statistically significant difference in phototrophs through the layers. The organisms that we did see shifting in terms of their guilds that were attributed to them through some literature searches from the metagenomic reads are the aerobes. So we think what's happening at 9.3 meters is that the oxygen concentration is really dominant here rather than light. And then at 9.8 meters, we see the dominance of this one organism, which is this green cyanobacterium called Formidium pseudopriestlii. And Formidium pseudopriestlii appears to be able to tolerate sulfide from some work done both physiologically with experiments and also by analyzing its genome. And so it's a combination of this organism being able to tolerate sulfide and anoxic conditions, as well as its propensity for photoautotrophy that allows it to live here and dominate this community. So a combination of light and oxygen at 9.8 meters appears to be structuring the community. We did, of course, also probe these mats for metabolic potential. So here I'm just showing this table that I used, that I generated to choose which genes to mark different pathways for metabolic composition and diversity. I tried to choose these genes carefully to represent metabolic processes of interest without over-representing any particular pathway. And the metabolisms that I chose to include here are photosynthesis, both oxygenic and anoxygenic, carbon cycling, including fixation, methanotrophy, and methanogenesis, nutrient acquisition, including polysaccharide degradation, nitrogen fixation, assimilatory nitrate and sulfate reduction, and anaerobic nitrogen and sulfur respiration, also phosphorus limitation. So when we examine the compositional differences in metabolism, we used this set of marker genes, and we found variation in different depths and mat layers, but very few statistically significant differences. So here on the x-axis is the relative abundance, and again, these samples are organized by water depth and mat layer. And phototrophy is here pictured in red. Blue is anaerobic respiration. This orange color here is a phosphorus limitation. Carbon cycling is in purple, and the nutrient acquisition is in green. And we did find some interesting trends and relationships here, including hints that nutrient limitation might be switching from nitrogen to phosphorus across the oxycline. We only found a couple of genes, however, that were statistically significantly different between our samples. So the first one is RBCL, which is a marker gene for Rubisco, and the second is PSBA, which is a marker gene for photosystem 2. So you can see here that the relative abundance of both of these genes decreases through the microbial mats at each depth, and that corresponds to the cyanobacterial abundances declining through the mat layers. So this was in support of the hypothesis that local light was shaping the community and pointing to cyanobacteria as a key community member controlling the entire microbiome of this ecosystem, which is not surprising. The fact that the cyanobacteria are so dominant also suppresses diversity, explaining some of the previous diversity results as well. So overall, the major result from this work is the support for meta-community theory. When we examine the mats at the meter scale, we see different controlling factors than when we examine them at the millimeter scale. Overall, the organisms in the mats in Lake Frixell appear to be spatially sorted fairly strictly according to these two dominant environmental habitat constraints, light and oxygen concentration. And there's one consideration that I speculate about a lot, but had no way of examining whatsoever. I really continue to wonder about how the populations of microorganisms shift seasonally, which, remember, is annually at the poles in this lake. We know from some shoulder season work done in the water column from the Priscu lab that the populations do appear to change, and there's evidence of mixotrophy in Lake Frixell as well. The temporal scales at the poles are extremely slow compared to other aquatic systems, which cycle daily and sometimes even faster. So the niches across these temporal scales may open and close throughout the year, and I think that's likely to allow for competition and coexistence of some species in ways that we really haven't been able to test yet. And this leads me to one of the last and most interesting results that I found, which is evidence of this marker gene for methanogenesis, HDRB. So HDRB is crucial to methane production, and it's strictly inhibited by oxygen. We found that HDRB homologs were found in statistically significantly higher relative abundance in the 9.8 meter surface samples, where the mats are anoxic only during the winter. And we found no evidence for methanogens in those samples. So this suggests that HDRB is hosted in non-methanogens. It's known to be in some sulfate-reducing bacterial genomes, and it might be necessary for energy generation in a wide variety of anaerobes. So in these organisms, HDRB is part of an enzyme complex that acts as an alternative to both substrate-level phosphorylation and electron transport, called flavin-based electron bifurcation. And this would be providing an alternate energy source in the winter when primary productivity through photosynthesis is not happening. So this was really exciting and a tantalizing result for me because it supported my very first hypothesis that photosynthetically active radiation, which is the energy source for the entire ecosystem, is limiting and dominating the spatial organization of these populations in the lake. There's a lot more to discuss and dig into here, but I'll end here for now. And thank you all very much for listening. And I'm looking forward to the remaining talk and the discussion after that.
Thank you so much. Very, very exciting and interesting. So but again, we will move on and come back to discussion at the end. Our next speaker is Wei-Dong Kong. So via Beijing, we're going to the Tibetan Plateau to examine bacterial diversity and network complexity. Wei-Dong.
Hello, everyone. I would like to thank Max for the invitation to discuss our work. My talk title is Bacterial Diversity and Network Complexity in Tibetan Plateau Lakes. I'm from the Institute of Tibetan Plateau Research, Chinese Academy of Sciences, which is based in Beijing. Okay, my talk includes 3 sections. The first, I would like just to briefly introduce Tibetan Plateau Lakes, and then brief introduction about the question we are interested in. And the last one is our research work. Okay, Tibetan Plateau is huge. It's around 2.5 million kilometers square in China part. And it's in the southwest of China. And the land area of the Tibetan Plateau is almost 1/4 of the European land area. Particularly, the Tibetan Plateau is controlled by westerly in winter, this side, bring some water, particularly some snow. And in the summer, the plateau is controlled by monsoon, mainly from the Indian monsoon and also mainly from the East Asian area. So this monsoon brings lots of water into the inland of the plateau, but in the western part of the plateau is very dry. So just because the plateau is very high, It's almost 4,500 meters high, and it's very strong UV and irradiance. So just because it's very high, it's very cold. The annual temperature lower than 0. And just because that, the summer temperature is still pretty low, just around 10 to 15 Celsius degree. So that means it's very cold. Also, it's very dry. Just I mentioned a little bit in last slide. Because it's very high, the monsoon couldn't bring many water into the inland. So the precipitation in the summer just around 40 to 800 millimeters. So the landscapes are very dry, including there are some meadow and steppe and many deserts too. The plant, just because it's very dry, the plant can grow very high, actually very short. It's just around several 2 or 3 centimeters. Just because of the very high and very cold, there are many glaciers on the plateau. Generally, it's around 10% of the global glacier mass on the plateau. Just because of the glaciers, the Tibetan Plateau is water of the water table of Asia. So the big rivers in Asia originally from the plateau, including the Yangtze River, the Yellow River, Yangtze River in China, and also many big rivers in Thailand and India. They are originally from the Tibetan Plateau. Just because of the many waters, so there are many lakes on the plateau. Actually, on the plateau, there are many studies in the lake microbial ecology, and some studies find the environmental filtering is a key factor controlling the microbial community. And also, a few studies, they find the microbial interactions also contribute greatly to the microbial community. Our studies on plateau lakes find that for the autotrophic microbial community, their diversity really controlled by the salinity. That means with increasing salinity, the autotrophic microbial diversity substantially decrease, which is common. Well, we didn't know that. Our results find that with the increasing nutrients, for example, the total organic carbon, the diversity substantially increase. That means the salinity decrease autotrophic diversity, but the nutrients offset the salinity constraints. So we are interested in which kind of factors really control microbial community, I mean the whole community in Tibetan Plateau lakes, for example, pH or salinity. And actually, we are really interested in the microbial interactions, how the microbial interactions change with increasing salinity. For this purpose, we investigated the microbial ecology in lakes across the whole plateau from the very west to the very east. Totally, it's around 25 lakes. We took the lake waters, and their salinity had a huge range from very fresh to very hypersaline. And we used the Illumina sequencing methods and a series statistic methods for this, including network analysis, random forest modeling, and correlations. Excuse me. Our data really show that the microbial diversity significantly decreased with the increasing salinity, which is common and very similar to our autotrophic diversity. Well, we find that the salinity really play a key role in controlling the bacterial diversity rather than pH, compared to the pH values and some other environmental factors. And also we compare the microbial community structure and also salinity really play a key role in driving the bacterial community structure. Well, in different types of the lakes, including freshwater, saline, and hypersaline, the key factors controlling the bacterial community a little bit different. For example, in freshwater, the water temperature is the key factor controlling the community structure. Well, in saline lakes, the salinity and the spatial distance play a key role in controlling the community structure. And also we calculated the core and the indicative microbiome. Very similar, the salinity play a key role in controlling the core and the indicative microbiome. So this really suggests the salinity drives both core and the indicative microbiome in the Tibetan Plateau lakes. And also we find a very similar story. The salinity changed the bacterial compositions. For example, with the increasing salinity, the alpha and the beta proteobacteria relative abundance significantly decrease the dark, the purple and the blue colors. The other way, for the gamma proteobacteria, which is the red, and for this dark yellow, there's relative abundance significantly increased. This suggests that salinity-driven environmental filtering really play a key role in changing their community compositions. As well as we calculated their correlations, our results find that for the positive correlations in freshwater, the alpha proteobacteria, this purple one, really dominated the correlations. Well, in saline and hypersaline lakes, the gamma proteobacteria really dominated the bacterial correlations in these lakes. For the negative correlations, very similar story. Also, we calculated the network analysis to see really what their interactions are. In freshwater lakes, the microbial interactions are relatively loose compared with the saline and hypersaline. The networks in saline and hypersaline lakes, their networks are very intenser compared with the freshwater. So this really suggests that the total correlations under their nodes gradually increase with the increasing salinity from around 200 to more than 900. Well, for the negative correlation, that means the bacterial competition in saline and hypersaline lakes, the competition are much stronger compared with the freshwater. So this suggests in hypersaline and saline lakes, the bacterial interactions are much stronger with very less niche differentiation. Summarize our story. Salinity substantially decreases bacterial diversity, and salinity changes the bacterial compositions, particularly the proteobacteria. And the most important we think is the salinity increases bacterial interactions by enhancing competition and meanwhile making their niche differentiation. Thank you.
Oh, thank you. Thank you, Weidong, and actually thank you, Martin, Marian, and Megan, all of you. This was really interesting, diverse habitats and questions. So we will open this up for questions and comments. And while I sort through what's come in on the chat, Martin, feel free to start, or anyone else if you have any questions for the others.
Yes, I have a question to Marian. You compared the diversity of the attached and the free-living bacteria in the water column. Could it be that the 16S rRNA sequences are not able to resolve some or more species among the free-living ones? Could it be that there's a trend? So to give you a little bit background of my question, I'm working on pelagic bacteria, free-living ones. Can you hear me?
I think so. I missed you for a moment, but I'm with you now.
Okay, so I'm working on planktonic bacteria, free-living ones, not attached ones, and this is a quite abundant group, Polyneucleobacter, and there we fail to distinguish species by using 16S rRNA sequences. So some species, they have completely, over the whole length of the gene, identical sequences. So we cannot resolve this important group. Could it be that this is a trend, that this is more frequent among the free-living ones, so they tend to have smaller genomes, smaller cell sizes, and probably the attached ones have larger genomes and are probably more diverse in their 16S rRNA sequences? Could it be?
Yeah, I think that's a really important point to mention. Of course, the measure of diversity that I used with the 16S gene is only as good as it is. And I think that a really exciting way forward, as you mentioned, is, you know, how representative are these, you know, short amplicon sequences of the total diversity that is present. We know that, you know, many different strains, as you mentioned, Martin, can be represented by the same 16S sequence. And so, of course, that is going to be masked in the data. And unfortunately, I'm not sure what the trend might be in terms of the relationship between, you know, the diversity of the 16S gene and how well representative it is of strain diversity, for example, in particles versus the free living. But I do, we delve deeper into these strains, and we'll learn more. So I do think that this is an important question that our field together tackles. And I think one thing that might also be interesting is that it's probably likely that the copy number variation of particle-associated bacteria for the 16S gene could be a little bit more variable than in the free living. And so the copy number is probably lower because, as you mentioned, Martin, the smaller genomes are much more streamlined in the free living. And so that's another layer that we don't fully understand because, of course, there could be variability within that copy number.
Thank you.
Okay, I have a question from the chat here. This is from Tulani Makalanyani and it's mainly to Wei Dong. In what way do the Tibetan Plateau lakes resemble climatically similar Arctic and Antarctic microbiomes?
Okay, that's a good question. Actually, it's I think the key point for the two poles because we call the Tibetan Plateau as the vertical. The temperature pretty low. And the second one is it's very dry in the two poles. They could be pretty similar. Actually, we had some preliminary data. We find that for example, in the deglaciated soils on the plateau, the microbes are very similar to Antarctic in terms of the general bacteria and also some autotrophic microbes. They are pretty similar, which really surprised me because in terms of the space, they are very different and it's very far. But their microbial communities are very similar. So I think in terms of the drought and the temperature, could be very similar in the two poles. But on the plateau, there are many vegetations too. So that's a big difference compared with the Antarctic.
Yeah, Megan, you want to comment? I guess not very much similar to the completely ice-covered lakes of Antarctica.
Yeah, right. So the ice covers do make a big difference for the McMurdo Valley lakes, definitely. So actually, when I was a postdoc at Berkeley Lab, I was studying Arctic soils. And so I learned a lot about the Arctic lakes up there that are thermokarst driven, right? So the effects of climate change have been warming the Arctic and then creating a landscape of these new lakes and their changes as they develop. I mean, they see huge changes in their carbon cycling, right? As they start to form and then sort of cycle through. And there's a lot, I think, more similarities between the Arctic landscapes and the lakes there and the Tibetan Plateau, specifically because a lot of the vegetation. So the Arctic has this huge gradient in vegetation from closer to the pole as you go farther south. And I know a little bit about the Tibetan Plateau. I would say it's more similar to the Arctic than the Antarctic for sure.
Okay. And jumping again around a little bit to something again, completely different question. And this is about the particulates and particles. This is a question on thoughts about whether changes in the biochemical composition of particulate organic matter has an influence on the bacterial diversity and the community structuring.
Ooh.
Mainly to Marian, but I think it's going to be a factor, of course, in the other systems as well.
So wait, it was the geochemistry of the particles and how it's going to influence the community composition. Oh, this is a fantastic question. And as you may have noticed, I took an aggregate of the community at 3 different time points through the year. And so I actually, with my study that I presented today, I cannot tease apart, this influence of the particle substrate and its influence on the community. But I think that there's a lot of really interesting research ahead of us where we tease apart these types of relationships. And I think that there's going to be a lot of influence on the community composition that the particle substrate has. So if the particle itself, an example from my talk was the pennate diatom, if it's something like that, then we can see that there's actually a symbiosis happening with the heterotrophic bacteria taking in photosynthate. But if it's a detrital-based particle, we would expect the habitat complexity to be much higher and support a much more diverse community that might tend to be more heterotrophic or create the opportunity for cross-feeding and cheating. And so here in a detrital-based particle or a photosynthetic particle, we would of course expect a dramatic difference in the community composition. And of course, I welcome more thoughts from the other panelists.
This is actually a follow-up on the number of bacterial OTUs that are unique to or shared between free living and attached. How does that look? I mean, the diversity in this is one, but how much is there actually crosstalk between those communities?
Ooh, that's also a really great question. We know that there's going to be migration happening as particle attached bacteria then break off and then go to find more particles. And so of course there will be kind of this relationship between the two. And also with the methods that I showed you today, there is a little bit of cross-contamination between, dislodging microbes on particles that then go to the smaller filter and vice versa. And so the methods that we have that I showed you today are a little incomplete in terms of really teasing that apart. But even with that cross-contamination, we can see unique organisms that live on particles that we miss if we don't sample those. And that's my broader overview of that. But
How do you see that, Megan, linking then to, I mean, particulate communities as biofilms versus then the much more complex microbial mats?
Yeah, I was interested if you sampled the free water next to your mats, Megan. Really interesting work.
Yeah, so no, we didn't sample the water column communities ourselves. There's actually been a lot more work done in the water column than there has been at on the microbial biofilms for just for logistic reasons, that it's a lot easier to drill a hole and throw a Niskin down in the water than it is to like melt a large hole and put a diver down there, right? So our team is really fortunate to have these biofilm samples, and we've gotten to piggyback off of a lot of the work that's been done previously with the water column community. So one of the starting places for hypotheses about the benthic microbial mat communities was the stratification of the communities through the water column. So there are some similarities and some differences, of course. So we do see, you know, differences in functional guild cell phototrophs, right, transitioning through the lake waters according to biochemistry of the water. One of the major differences that was interesting to us was the lack of anoxygenic phototrophs in the benthic microbial mats, which are definitely available in the water column. And one of the reasons we think that is the lack of appropriate wavelengths for the anoxygenic phototrophs that I was mentioning at the very beginning there. That being said, I expect there's a lot of communication between the water column communities and the microbial mat communities. And that's one of the things I would love to, you know, if I had infinite lifetimes, chase down all these questions, right? So yeah, definitely it would be interesting to look at organisms traveling between these two communities, presumably seasonally as resources change. Yeah, I think that would be really interesting. But no, I don't have a direct answer for how that relationship looks right now either.
Okay. So another question, this is from Marinella Laporte, that gets us back to the Tibetan Plateau and the effect of salinity. And so Weidong, you examined this from the bacterial diversity, but of course salinity is going to affect animals. And the question here is, would salinity also interfere with the microbiome diversity of invertebrate-associated bacteria? And could this be something of the bind that it's actually the microbiomes on animals that's also playing a role?
Yeah, that's a great question. Actually, there on the plateau there are many animals, many wild animals, as well as human-managed animals like cattle and sheep. There are many, but relatively they're much less compared with the like the plain, like the Beijing area. That's a good question. I do believe the animal microbiome could contribute to the lake. That's very possible, but as I know, no studies about that.
Okay, how is your connectivity to the glaciers or turbidity gradients in lakes also that you have? So is that an examination that you've done?
Yeah, we did that. I think last year or 2 years ago, we had a paper, possibly in, I think it's in the FEMS Microbiology Ecology, possibly also handled by Martin. It's you. But anyway, the general answer is there's a huge difference from the ice microbes and the water from the melting water from the ice, and particularly compare with the microbes in deglaciated soils and ice microbes and the lake microbes. There's a huge difference in lake. So we think the lake could be a very important ecosystem. I mean, change the microbial community originally from the deglaciated soils as well as the melting waters. Very less connectivity compared with the lake microbial community.
Okay, any final comments or questions? We're getting into an hour and a half, so I think we're going to have to wrap up. There are a few more questions still here, so we will share these with the speakers, and we can then get back to you on by email. So I definitely want to thank Martin, Marian, Weidong, and Megan. Thanks for taking part in this webinar. It was really interesting hearing from all of you, and thank you to everyone in the audience for attending and for your questions. And we will meet again for something completely different in a month on November 12th about the soil microbiome. So again, we're going to look at the ecology of soil microorganisms on Thursday, November 12th, with a new series of talks. So again, thank you very much, and take care, everyone.
Okay.
Thank you so much for having us.
Good working, everyone.
Bye.
Bye.